This is a video tutorial for Curtin, the proteomic data analysis tool brought to you by researchers from the MRC Protein Phosphorylation and Ubiquitination Unit at the University of Dundee. This project is supported by the Aligning Science Across Parkinson's Research Network. Hello Curtin user, and thank you for your interest in this video. In the next few minutes, I'm going to show you how to upload your proteomics data into Curtin using example data from the Curtin publication. You will see here that you are required to upload two files, a differential analysis file and a raw file. You then need to specify the appropriate columns from these two files in the drop-down menus below, before clicking Submit and visualising your data. So the differential analysis file, I'll just show mine for illustration. So this needs to have the fold change and the significance of that change between the two conditions that you want to compare in Curtin. So this could be an output from Perseus, for example, or MS Stats. But the raw file, as you see here, this must have your unaltered intensities from whichever software you use to search your data. So this could be an output file from Spectronaut or MS Fragger, MaxQuant or Diane, for example. And this file is usually exported in a tabulated text format like this. But you might want to make some alterations to the file in Excel before uploading to Curtin, such as deleting unnecessary columns or changing the column headers to simplify them. And this is fine as long as you remember to save a copy of your modified file as a text file, which is very easy to do in Excel, just like this. Just making sure that you choose the .text format to save your file, because you cannot upload these Excel workbook files into Curtin. So to upload your file, just click Choose File, select the appropriate one, and wait for it to appear in the white box. Then pick your raw file. Good, and now they've both appeared. It should be possible to select column headers from those files in the drop-down menus. So for this first option, select primary IDs, it's best to select the column in your file with the UniProt IDs to get the most out of Curtin, because it will then extract data about each of the proteins in your dataset from UniProt as long as this box here is ticked. So in my file, this column is called index. So I will select that here. For the next drop-down option, select fold change. Pick the column with the fold change between the two conditions that you want to compare in Curtin. These values are already log2 transformed, but it's fine if you just have the untransformed fold changes, because you can select this box and Curtin will perform that transformation for you. So I'll pick that column. If you were to select this box, then it would in effect reverse the direction of your volcano plot. For the third drop down, select significant, choose the column in your file which has the p-values. So again, I think mine are already negative log 10 transformed, but if you just had the untransformed p-values, you could select this box and Curtin would perform that transformation. So I'll just select that column. The comparison drop-down, it's useful if you have multiple differential abundance comparisons in your file. For example, here I have two time points, 4 hours and a 24-hour time point, and I might want to be able to switch between looking at data for those different time points in the same Curtin session, but you don't have to select anything for this drop-down, and I won't this time. So then for the raw file, select primary IDs. Best to just pick the same column header as you used for your differential analysis file. So again, this just has the UniProt IDs. So in my file, that's called index again. So that's what I've selected. And then for select samples, you'll notice that this allows multiple selections. So just choose the relevant experimental sample identities. So I'll just make a multiple selection here. And now with everything selected, we are ready to click submit. Before we do, I would just like to show you how to add any metadata associated with your experimental data. So you might notice this format is the same as that for the PRIDE database, but it just allows you to add some more contextual information for the data. So for example, what organisms the experiment was done in, any diseases it's relevant to, cell types, protocols for the sample processing, the data processing, which MS instrument it was run on, methods, any PTMs detected, tags, software, affiliations, authors. So we do recommend that you include as much additional information as you have. So I'll just close that. And now we are ready to click submit. So you can follow this blue bar below to check progress. And there are various factors that might affect the speed, so internet connection, speed, file size, they'd be the main ones, but it shouldn't take more than a minute to process. So now that it says finished, you can scroll down and you'll see that the volcano plot has been plotted. We won't go into exploring that in this tutorial, just checking that it's there. But I will just show you how to save your session, because we recommend that you do that at this point, really just after submitting your data, before you've selected any proteins on the volcano plot or started to do anything like that. So just go to session, save session, and wait just a few moments, and then you'll see a link appear at the top, and also a link to a QR code, which you can also use. So this is a shareable web link, so you can copy and paste this to share with supervisors, collaborators, or just save it somewhere so that you can use it to re-access the session yourself. So thank you very much for watching this tutorial. Please have a look at our other videos to learn how to visualize and explore your data.